Okay, today we'll be revising IASM block number 24, which talks about blood cells, haematopoiesis, the formation of blood cells, their histological features and functions. So first of all, in a blood test, in a blood sample, there is composed of plasma, which makes about 55% of it. It's the mainly water, liquidy part of the blood. Then we have the buffy coat, which contains white blood cells and platelets. Platelets are blood fragments. Leukocytes are white blood cells, which contribute to the immune response. Then we have 45% of the blood, which contains the erythrocytes, which are just red blood cells. Together, these white blood cells, red blood cells, and platelets are the formed elements of the blood, which we have to make. So the blood plasma, going through really quickly, is just the solvent part of the blood. It contains proteins and other solutes. The proteins contain such as these coagulation factors to clot blood, and solutes, we have electrolytes and nutrients which help maintain bodily homeostasis. Usually in blood tests, you just use the serum, which is just blood plasma but without all the clotting factors, the coagulation factors, and you can do that by either adding anticoagulants such as citrate, or you can just let it clot and then remove the blood clots via centrifuge. So the three formed elements, as we mentioned, we have red blood cells, also known as erythrocytes. They're the main components within blood. Then we have white blood cells, and here we can talk about these leukocytes in more detail later. Then we have platelets. So first thing we need to understand is that the process of forming blood is known as haematopoiesis. So hemato is blood, poiesis is forming or making. And this always occurs in the bone marrow. So the two main things you need to know about haematopoiesis is that it comes from pluripotent stem cells. You don't need to know all these intermediates, but mainly these two: lymphoid stem cells and myeloid stem cells. So myeloid stem cells makes all of these: the red blood cells, platelets, eosinophils, basophils, neutrophils, and the monocytes. Then we have the lymphoid stem cells, which make these types of lymphocytes, which are T lymphocytes, B lymphocytes, and natural killer cells. But just know that these come from lymphocyte stem cells, and these come from myeloid stem cells. So, blood formation occurs really early in the embryological development. At 3 weeks, it forms in the yolk sac of the embryo forms these blood islets. At 4 to 5 months, the liver and the spleen is in the dominant place for haematopoiesis. And then at 8 to 9 months, when the bone marrow forms, it predominates in the bone marrow for blood formation. You can see that the infant has a lot of red bone marrow, and it forms a lot of blood everywhere in the body. However, in adults, haematopoiesis only occurs in the axial and the proximal ends of long bones, such as the femur. And then this left femur. Then bone marrow examination. It's done by taking aspiration and then examining the tissue to diagnose some hematological diseases, such as aplastic anemia, which is when your body stops making new blood cells, and leukemia. So the places where you can extract tissue is the posterior iliac crest, which is here. Or you can take it from the anterior iliac crest, which is here. And this is mainly when you can't access these places because the person is obese. Or you can also get it from the sternum, which is here. And that's only when the iliac crests, both posterior and anterior, so the front and the back, is not accessible. And the tibia, which is this bone right here, this bone, this on the inner side of the leg, you use it to extract tissue in infants only. So moving on, erythropoiesis. Erythro is red blood cell. Poiesis is making, the development of red blood cells. So you start with a pluripotent haematopoietic stem cell, as we saw here. And then what happens is that there is a growth factor that you have to memorize. It's called erythropoietin, and it's a glycoprotein hormone which regulates the formation and development of red blood cells. All of this happens in the bone marrow, and you need to understand some of the morphological changes that occurs throughout the development. So the stem cell, after it gets triggered by the growth factor, it will form a proerythroblast, and usually when the words have blast in them, it means it has a lot of these stem cells which allow development. So as you notice, as it goes down the development chain, the cell size gets smaller and smaller, and the cytoplasmic volume, so this part, decreases, and the the cytoplasmic volume increases, but the nucleus decreases until there's none. And when there's none, this happens in the peripheral blood, which means that the erythrocyte is mature. And something to notice is that the staining changes from eosinophilic staining, so this blue purple staining, to just plain red it changes from this blue-purple staining, which is called H staining, and this red is the eosinic staining. This is because H staining, it stains all the DNA and the nucleic acids and nucleic material, whereas the eosinic staining stains proteins. And it's red because of the accumulation of hemoglobin. Okay, some of the histological and morphological features of red blood cells and platelets. First of all, it's a biconcave shape. This is because we need to maximize the surface area of the red blood cell for the efficiency of substance exchange. Then we have a very strong and flexible plasma membrane, so it allows it to squeeze through narrow capillaries without breaking or rupturing. Then it's non-nucleated, as the development showed that the nucleus just vanishes to make more space for a cytoplasm. So the blood cells cannot regenerate other blood cells. They have to be formed by haematopoiesis in the bone marrow. So they can't reproduce themselves, and they cannot carry out metabolic activities. They have a high concentration of hemoglobin in the cytoplasm, which firstly makes it red, and also allows oxygen and carbon dioxide to bind to it, so it can be transferred and transported throughout the circulation. And also, when we look at microscopic pictures of blood, we use red blood cells as a histological ruler, because they are almost always, they're just 8 micrometers. So in 8 micrometers, we can just make measurements throughout the image. So something you need to know is the regulation of red blood cells and also the destruction of red blood cells. So when you're asked to give a example of a negative feedback loop, you can always talk about the regulation of red blood cells. So if you're bleeding a lot, or something happens that results in low oxygen levels or low blood levels, this is known as hypoxemia, and this alerts the liver and the kidneys in which they, after being sensed by the liver and the kidneys, they increase the secretion of EPO, erythropoietin, which was the glycoprotein hormone that stimulates red blood cell production. And then this protein hormone will then tell the red bone marrow in the iliac crests or other places, red bone marrow, to increase the development of red blood cells. So from a proerythroblast to form a mature erythrocyte. And once after 3 to 4 days, the increase in red blood cells will then allow more O2 transport, and then this will not decrease the secretion of EPO, as you don't need as much synthesis of red blood cells, formation of red blood cells. Okay, and talking about the destruction of red blood cells, actually they have a lifespan. The erythrocytes, they die every 120 days. But then how red blood cells are degraded is that it forms two groups, the heme group and the globin groups. The globin groups are simple. They just get hydrolyzed into free amino acids, and then they can be taken up by the body for uses or storage. The heme group, it makes biliverdin and iron. Iron can be stored in the body, reused, or lost during menstruation or in injury. Biliverdin thus gets converted into bilirubin, and bilirubin can be converted into bile and excreted through feces. If there is excess bilirubin in the blood, bilirubin is a yellow pigment, and this yellow pigment, if it's in the blood for a prolonged period of time, it causes jaundice. Jaundice is the yellowing of the eyes and the skin, and often occurs in infants because bilirubin is used to break down red blood cells. So if they can't be excreted, then your body will turn yellow. Then let's talk about platelets. So the importance of platelets, to understand, the main point is that they're not cells and they're fragments, and they are formed via fragmentation from these megakaryocytes. So they pinch off like this, and they form these tiny platelets, and they are stimulated by thrombopoietin to make more platelets. So some morphological features about these platelets are they're small, very small in comparison to a red blood cell, which is 8 micrometers, and they don't have a nucleus because they aren't cells and they're just bits off of a megakaryocyte. Their function is for hemostasis, which controls bleeding. So what they do is that they survey the endothelial lining of blood vessels for any injuries or breaks, and then they'll form a platelet plug on the damaged site, and the degranulation of platelets will release glycoproteins that signal for vasoconstriction, so then there won't be as much blood flow at the site of damage. And it also facilitates blood clots, so you make a plug and then a clot. After you form the clot, the platelets also help in clot retraction, which pulls in the walls of the blood vessel, and then the blood vessel is fixed. Moving on to leukocytes, or white blood cells, we're only going to be talking about myeloid stem cells, the leukocytes formed by myeloid stem cells, which are neutrophils, basophils, eosinophils, and monocytes. Lymphocytes are from the lymphoid stem cells. Yeah, so leukocytes, they provide protection against infections and other diseases. So it contributes to our immune system. So they can be separated into 2 types. So we have 1, 2, and 3. These are the granulocytes. They have granules, as you can see the dots. inside. Then we have the agranulocytes, which are the lymphocytes and the monocytes. So, neutrophils are the most common in circulating leukocytes, contributing 50 to 70%. Their key features are that, firstly, they are multilobulated. You see these multilobulated, 1, 2, 3, 4, like that. And they're connected by strands of chromatin, like so. And they have, as they grow more mature, there are more lobes. And in females, if you see these tiny little dumbbell-shaped things protruding out of the lobes, they are called bar bodies, and they're condensed X chromosomes. And a thing to note about this cytoplasm is it's relatively more clear and neutral, and these are non-specific granules. So the function of neutrophils is phagocytosis and chemotaxis. Chemotaxis is to help move cells towards the infection site. Phagocytosis helps to degrade bacteria by engulfing the bacterium. Next, we move on to eosinophils. So eosinophils make up 1 to 4% of the circulating leukocytes. The morphological features we talk about nucleus first is that it is bilobed, so 2 lobes, 1, 2, 1, 2. Usually it's bilobed. And in the cytoplasm, you will see a lot of eosinophilic stained specific granules. So the eosinophilic stains protein granules, so you'll see a lot of red dots and granules in eosinophils. So this is quite easy to remember because the name eosinophil and eosinophilic granules. The function of these are to protect against parasitic infection and to modulate inflammatory responses that are triggered by allergies. So in a blood-stained sample, if you see a lot of eosinophils, then maybe this person is undergoing a parasitic infection or they are having an allergic reaction. And next, we talk about basophils. Basophils are the least amount of circulating leukocytes, less than 1%. So their nucleus is also kind of bilobed, although it is clearly not as clear as the eosinophils. They are known as obscure, unclear structure. Their cytoplasm is filled with not eosinophilic granules, but basophilic granules, so you can link that with the name. So the basophilic granules are stained blue-purple, these. Their function is to aid the mobility and the action of other leukocytes during an allergic response. And their granules, their basophilic stained granules, release histamine, which is a vasodilator. It opens up the blood vessels, and heparin, which is an anticoagulant. Next, we have monocytes. These are agranulocytes, so no granules. They make up 3 to 8% of the white blood cells, and they are the largest in size. So key features are that its nucleus is kidney-shaped or horseshoe-shaped. And they rarely contain granules, but they have some azurophilic granules. But how you remember this is that they kind of look like bubbles. They look frothy and foamy, the cytoplasm. And these monocytes are precursors for the tissue macrophages or other phagocytic cells in other tissues. So in the liver, the macrophages are the Kupffer cells. In the nervous tissue or in the brain, it's the microglia. In the bone tissue, it's the osteoclasts. So these form the precursor cells. Lastly, we are going to talk about lymphocytes, which make up 20 to 30% of circulating leukocytes. Their structure is that their nucleus occupies a lot of the volume of the cell, 95% of the cell, and it's super darkly stained. And it has a very thin ring for its cytoplasm. So these are its distinct features. The function is to regulate cells' immunity. So the B and T lymphocytes, as we mentioned before, they mediate an immune response, and the natural killer cells can kill invading target cells. So the summary here is that you can split these leukocytes into two big groups, the ones with granules and the ones without granules. The ones with granules, we have neutrophils, basophils, and eosinophils. Neutrophils have clear or neutral staining cytoplasms. Basophils have basophilic granules, which are red. They're blue. Then the eosinophils have eosinophilic granules, which are red. Sorry, I'm stupid. Basophilic is blue. Eosinophilic is red. And all three of these this is multilobed. This one is bilobed. This one is bilobed. Here, so bilobed eosinophils, bilobed basophils, but obscured. Neutrophils work in phagocytosis, actually ingesting the bacterium and guiding the cells towards the site of infection. Basophils help other leukocytes in the allergic response, and their granules release histamine, a vasodilator, and heparin, an anticoagulant. Eosinophils also modulate allergic responses, but they are more specified in protection against parasitic infections and also allergic reactions. The agranulocytes, no granules, they are the monocytes or the lymphocytes. The monocytes and the lymphocytes are monolobed. The monocytes have a frothy or foamy kind of cytoplasm with zero to little no granules. The lymphocyte has a thin-rimmed cytoplasm with a huge nucleus. So monocytes are precursor cells for tissue macrophages, microglia in nervous tissue, Kupffer in liver cells, and osteoclasts in bone. The lymphocytes help mediate immune response, the B and T lymphocytes, and whereas the killer cells they kill target cells. Whew. So now we're gonna be trying to identify some different blood cells under a microscope through a blood smear. So we have the methylene blue stain, which makes it blue or purple, and it stains DNA and RNA. So any genetic material is stained blue, so the nucleus. Then we have acidic dye, which is the eosin. It makes it red and pink, and it stains proteins. So here we can tell that this is a lymphocyte because its features are it has a humongous 95% nucleus. Here, these are platelets because they're cell fragments. These are red blood cells. A lot of these are red blood cells. A neutrophil because it's multilobed. You see these purple bits multilobed. This is also a neutrophil. Then, here, neutrophil again, platelets. Lymphocyte, look at the big nucleus, another neutrophil here. A lymphocyte, a monocyte because of its kidney or horseshoe-shaped nucleus. Then we have these individual platelets. Then we have a neutrophil, which happens to be bilobed and connected with chromatin. This one as well. And these are just red blood cells.